Hello everyone, I hope you are doing well. So in this video we're gonna talk about how to find the promoter sequence of a gene in NCBI. And first of all, let's have a review of the transcription mechanism. As you know, we have two different strands on the DNA. One of them is the strand from 5' to 3', and the other one, which is complement to this one, is from 3' to 5'. And if we consume that this is our gene sequence, the upstream sequence of our gene sequence is called promoter. The strand which the gene sequence exists, we call it coding or sense strand, and the other one we call template or antisense strand. And as you know, the transcription factors and RNA polymerase actually identify the promoter sequence, and they actually create a complex and start the transcription. If we can assume that this is our mRNA, the direction for transcription is from 3' to 5', and this is the 5' end of our mRNA, and this is the 3' end of our mRNA. And as you know, the mRNA is a complement to the template strand, and it actually is making based on the template strand. I mean, the RNA polymerase use the template strand to continue transcription and to make the mRNA. But the sequence of mRNA is exactly similar to the gene sequence, which is on the coding or sense strand. And as example, as you see, this is our DNA sequence, and the upper strand here is coding strand, and this is template strand. As you see, the RNA is making based on the template strand, and the transcript or RNA is similar to the coding strand. Of course, as all of us know, there is a difference. RNA has U and the DNA has T. And we can take a look at actually another example here. As you can see, this is 5' to 3', our coding strand or gene, and this is our template strand, and RNA polymerase is making the mRNA based on the template strand from 3 to 5' based on the template strand. So there is another DNA strand classification. We talked about the coding and template strands, but we also have forward strand and reverse strand. The forward strand is always from 5' to 3', and the reverse strand is from 3' to 5'. And in the important point is that we always see the sequence in most of the bioinformatic websites. We see the forward strand in on bioinformatic websites like NCBI and other websites. So this is the second DNA strand classification, and we have two kind of the position for the coding and template strands. One of them is when the coding strand is on forward strand. As we saw in the first slide, this is an example when we have forward strand here and reverse strand here, and coding strand or our gene is on the forward strand, as you can see, and the reverse strand is here, and our template strand also is on the reverse strand. We always have forward strand. The upper strand is always called forward strand, and the lower strand is always called reverse strand. But the coding or template strands can be replaced, as we will see here. The coding strand is on the reverse strand. This is our forward strand, and this is our reverse strand. And our gene sequence now is on the reverse strand, and this is our promoter, because our promoter is on the 5' end of our sequence. This is our promoter strands, this is our gene sequence. The transcription factors identify the promoter sequence and helps RNA polymerase to stick to it, and they start the transcription based on our template or antithesis strand, and this is our direction. For the template strand is being read from 3' to 5', and the mRNA is being created from 5' to 3'. So the first condition when we want to retrieve the promoter when coding strand is on forward strand, and this is the simpler one, because we can simply we see this actually box in NCBI, and it shows the number of nucleotide, the first nucleotide on the gene, and this is the last nucleotide, the number of last nucleotide on the gene, and this is from 5' to 3'. So this number is the number of nucleotide here, right here. This is right here. So if we want to retrieve the promoter sequence, we just need to go backward, go reverse to get the promoter strand. So if we want to have the number of nucleotide, we just need to easily subtract this number, whatever number we want. For example, 100, 1000, 500, whatever we want. And if we subtract a desired number, we can get the promoter sequence. We can go reversely, we can go backward to get the promoter sequence. So we can easily get the promoter sequence by subtracting this number from the 5'. But the other condition we have when the coding strand is on the reverse strand. We need to know that, as I told you, always we see the forward strand sequence in NCBI. This is the sequence when we retrieve a sequence of a gene. This is what we see in the NCBI. So if we wanted this sequence, this is our promoter sequence, we first need to desequence the complement sequence and somehow convert it to the promoter sequence. So we need to this is actually this number is the number of nucleotide here, right here, and this is right here. So if we want this sequence, we just need to add the desired number to this number, so we can go forward and get the sequence of actually this area. So if we add desired number, we get this sequence, which is right here. And if we want to get the promoter sequence, we need to activating the reverse complete option. There is another box below this box in NCBI, and we just need to actually activate the show reverse complement in NCBI, so that we can get the promoter sequence. And if we do that, we can get the promoter sequence. And one super important point, guys, all of the FASTA formats are from 5' to 3'. We don't have any FASTA format from 3' to 5'. So if we get this sequence right here, if we want to just make it complement, we will have this one, I mean TCGCA, but this is from 3' to 5'. We just need to reverse it. First we need to complement this sequence. We get this sequence, and we need to reverse it to get this one, because we need the sequence from 5' to 3'. And I will explain it more. Let's actually can assume that this is our sequence, which is this sequence, the complementary sequence of our promoter sequence. So this is our sequence from 5' to 3'. If we create the complement sequence, will be this, as you know, from 3' to 5'. And if we get the reverse strand, we will get this one. I actually reversed it. We will have GATTG, GATTG, as you know here, from 5' to 3', 5' to 3'. And if I write it in the right font, we will have GATT, as you know, GATTGC. So this is our promoter sequence, because as I told you, it is always, we see always faster formats in 5' to 3'. There is no faster format from 3' to 5'. So when our gene is on the reverse strand, we need to get the complement reverse strand to get our promoter sequence. So this is up to here, and we're gonna look at a couple of examples so you can get the actual mechanism how to retrieve the promoter sequence from NCBI. So I want to practically show you how you can retrieve the promoter sequence of a gene. I have this gene COX4I1. This is a gene related to human genome, and we want to get the promoter region of the gene. Very important point is that this arrow shows us if our gene is on the actually forward strand or reverse strand. If this is from left to right, this is a gene on the forward strand, and I have another gene, EMC8. As you can see, this is from right to left. The arrow is from right to left, so this is on the reverse strand. So let's first get the sequence of our gene on the forward strand here. But before that, let me show you. You can select the gene section of NCBI and write your desired gene, and get the you will be directed to this page, and you can see all of these options. In genome regions, transcripts, and products, you can get the FASTA sequence. So now I have the FASTA sequence of my gene. So as you can see, we have this box, and we have another box here. We don't need to activate none of them. But for example, because as we saw, this gene is on the forward strand, we need to subtract desired number to get the promoter sequence. For example, I wanna the 500 nucleotide upstream from my gene, so I easily subtract 500, and I can write it to 1. So right here, the first approximately 500 nucleotide from the in this actually sequence is my promoter, are my upstream region of my gene. And we can check it with this website, eukaryote promoter database. I have searched the COX gene, and you can find your gene from in NCBI with this link. And here, this is this option sequence retrieval tool, you can, for example, this is from minus 499 to the 100 on the gene. So you can get the sequence and you will see this. And if I, for example, copy this and search it here, as you can see, this is the promoter region of my gene. I can do it again. As you can see, this is the sequence from minus 499 to 100 nucleotide, and this is the all sequence of the gene plus the 500 nucleotide upstream of the gene. So we easily retrieve the promoter region here, and let's take a look at the other example, the EMC8 gene. Here, as you see, this is on the reverse strand, and I want to get the fast sequence. So first, let me deactivate this. So as we saw, we need to just add desired number to this number to get the promoter region. So I've added 500 to this number, and this is the result. And I need to activate the show reverse complement, and we need to update the view, and this is the first 500 nucleotides or the upstream region of our gene. And we can here in EPD website, we can go to this part and, for example, get the upstream or promoter region, and exactly as you see, this is the promoter region of our gene. Thank you guys for paying attention to this video. Please subscribe to the channel and like the video if you liked it, and also comment anything you want and suggest any topic you wanted me to make for you. Thank you very much. See you later. Bye.